In this video, we're going to wrap up the material for Chapter 25. So by the end of this lecture, you should be able to say that you can distinguish between disaccharides, oligosaccharides, and polysaccharides, explain how carbohydrates are involved in signaling between cells using antigens and antibodies, explain how glycolipid antigens are anchored to the cell membrane, and appreciate the role of polysaccharides in familiar contexts. Most carbohydrates in nature contain more than one monosaccharide unit. Those that contain 2 units are called disaccharides. Those that contain 3 units are called trisaccharides, and so forth. The general term oligosaccharide is used for carbohydrates that contain from 4 to 10 monosaccharide units. Carbohydrates containing larger numbers of monosaccharide units are called polysaccharides. In a disaccharide, 2 monosaccharide units are joined together by a glycosidic bond between the anomeric carbon of one unit and an OH of the other. 3 important disaccharides are sucrose, lactose, and maltose. Sucrose, which is table sugar, is the most abundant disaccharide in the biological world. It is obtained principally from the juice of sugarcane and sugar beets. In sucrose, you can see that the glycosidic bond is formed between the anomeric carbon, or carbon 1, of glucopyranose, whereas it's joined to carbon 2 of the anomeric position on fructofuranose. This linkage between position 1 and position 2 makes it a 1,2 glycosidic bond. And then the fact that this stereochemistry here is alpha, it's opposite to this CH2OH group here, tells us this is an alpha 1,2 glycosidic bond. It's kind of a mouthful to keep saying glycosidic bond, so people instead just say it's an alpha 1,2 linkage. Lactose is the principal sugar present in milk, and it has a beta-linked 1,4 glycosidic bond. Maltose derives its name from its presence in malt, which is the juice from sprouted barley and other cereal grains. The alpha linkage here at carbon 1 is attached to position 4, and so this is an alpha 1,4 glycosidic bond. Although all monosaccharides are sweet to the taste, some are sweeter than others. Here's something fun, a sweetness index. This explains why the artificial sweetener packets always seem to be basically empty and yet make your coffee utterly undrinkable. Membranes of animal plasma cells have large numbers of relatively small carbohydrates bound to them. In fact, the outsides of most plasma cell membranes are literally sugar-coated. These membrane-bound carbohydrates are part of the mechanism by which cell types recognize each other and act as biochemical markers. Typically, these membrane-bound carbohydrates contain between 4 and 17 monosaccharide units. Among the first discovered and best understood of these membrane-bound carbohydrates are those of the ABO blood group system, which was discovered in 1900 by Carl Landsteiner. Whether an individual has A, B, AB, or O blood is genetically determined and depends on the type of trisaccharide or tetrasaccharide bound to the surface of a person's red blood cells. The monosaccharides of each blood group and the type of glycosidic bond joining them are shown here using the shape and color for representing glycans. People with type O blood, such as myself, are referred to as universal donors because their RBCs don't present a galactose or a GalNAc as found in AB blood. Therefore, antibodies that target these extended antigens are not reactive to the H antigen in type O blood. Meanwhile, type AB blood donors have both of these additional sugar units, but no antibody in their plasma, and they're known as universal acceptors. The 3 blood type antigens are drawn out here, and you can see the additional galactose in the B type antigen has an OH at the 2 position, and the GalNAc in the A antigen is also galactose based, but it has an N-acetyl group at the 2 position. Then down here you can see drawn out the O antigen, and this part of the structure is conserved across all the antigens. These antigens are linked through a short linker to a lipid that's known as ceramide. The ceramide intercalates into the cell membrane and then serves as the anchor to attach the glycans to the cell surface. Starch can be separated into two principal polysaccharides, amylose and amylopectin. Complete hydrolysis of both amylose and amylopectin yields only D-glucose. Amylose is composed of unbranched chains of many glucose units that are joined by alpha-1,4 linkages, and the alpha-1,4 linkage is what actually causes the amylose to take on this kind of helical shape. Amylopectin contains chains of alpha-1,4 glycosidic bonds and alpha-1,6 glycosidic bonds that are combined in this branch-type structure shown down here. Cellulose, which is the most widely distributed plant skeletal polysaccharide, constitutes almost half of the cell wall material of wood. Cotton is almost pure cellulose. The cellulose is a linear polysaccharide of glucose units joined by beta-1,4 glycosidic bonds. Cellulose molecules act like stiff rods, which is a feature that enables them to align themselves side by side into well-organized water-insoluble fibers, in which the hydroxyl groups form numerous intermolecular hydrogen bonds. This arrangement into bundles gives cellulose fibers their high mechanical strength. It's also the reason that cellulose is insoluble in water. When a piece of cellulose-containing material is placed in water, there are not enough interactions with the water molecules on the surface of the fiber to separate individual cellulose molecules away from the strongly hydrogen-bonded fiber. Glucosaminoglycans are a group of linear polysaccharides composed of repeating disaccharides in which one of the monosaccharide units has either a negatively charged carboxylate group or a negatively charged sulfate group. Members of this family of carbohydrates include hyaluronic acid, heparin, chondroitin sulfate, and keratin sulfate, which are components of cartilage, tendons, and other connective tissues, and dermatin sulfate, which is a component of the extracellular matrix of the skin. A general characteristic of this class of polysaccharides is a repeating disaccharide consisting of units of uronic acid and an aminohexose with a 1,4 glycosidic bond between the aminohexose and the uronic acid. Hyaluronic acid is present in connective tissue. It's the most abundant in embryonic tissues and in specialized connective tissues such as synovial fluid, the lubricant joints in the body, and the vitreous humor of the eye, where it provides a clear elastic gel that maintains the retina in its proper position. Heparin is a heterogeneous mixture of variably sulfonated polysaccharide chains that is synthesized and stored in mast cells of various tissues, particularly the liver, lungs, and gut. Heparin has many biological functions, the best known and understood of which is its anticoagulant activity. It binds strongly to antithrombin 3, a plasma protein involved in terminating the clotting process. Heparin is used medically as an anticoagulant.